thanks again for the introduction, and also I would like to thank Prof. Chow and the AHCC trial group invitation for inviting me to present this in this afternoon. So I will try to keep it short because I know that after my talk will be the break. So I will promise to keep it short. So this afternoon, my topic is to present or share with you some of the data that we have done for the Y90 treated patient. So I just want to say that I don't have any conflict of interest, but we are really interested in analyzing this data, and there are a lot of collaboration. But the outline of my talk today, I will just briefly touch on the SERT treatment, and also share with you some latest data that we have discovered, and also some immunological research for the responder of SERT treatment. So I always have the feeling that this group is a well-trusted community. So I will be presenting all the data that are unpublished or just submitted for manuscript. So for the SERT is selective internal radiation therapy. So I'm not an expert in this area, but the idea is that the radioactive beads will be delivered to the tumor region, close to tumor regions. Then the beads will go into the tumor and then kill the tumor by the radiation. And according to the management of the locally advanced HCC published by Prof. Chow earlier, so it's quite common to use a SERT as the treatment for the locally advanced HCC. And also because of this, then we are interested to ask a question, are there any predictive markers or any form of biomarker we can identify from the patient even before the treatment of SERT, which might be potentially predictive for the treatment outcome? So, and also thanks to other collaborators, so we embarked the journey of translational analysis and also the biomarker discovery for SERT treatment. So for the first part, I want to touch on the trial that was led by a German team from Prof. Jan Riekers. So we focus on a trial called ceramic trial. So this trial, cut the long story short, is a phase 2 randomized controlled trial. The main idea is to compare the SERT plus sorafenib versus sorafenib alone in this patient. And then for this patient, the target cohort are those who took part in the palliative cohort of the ceramic studies, and they are not eligible for resection or ablation, or even they are not good candidate for TACE treatment. Then from this, you can see the diagram. Actually, it's a randomized trial. So some of the patients will go through sorafenib alone treatment, and some will go for Y90 treatment first, followed by the sorafenib treatment. Then this is those are inclusion criteria and all the information for the eligible patients. Then the key findings from this clinical trial is that unfortunately there is no significant improvement in OS compared to sorafenib alone. So the KM plot here is showing that there is no overall survival benefit, but it's a positive signal in the way that there are similar safety and toxicity profile. And also, based on some subgroup analysis, actually we can identify some clinical benefit from those patients who are younger. In this case, is younger than 65 years old. And also, we do see the clinical benefit from those patients with non-cirrhosis and non-alcoholic etiology. Then we keep asking a question, like when we collaborate with this group of people, then we keep asking what type of translational research we can do, and what type of biomarker we may be able to identify. But just give you a brief background information before we initiate our studies. Actually, this group of people, they are very productive. They have been analyzing a lot of different data. So for example, they have been looking at some clinical signals. They published few papers and proving that the MVC's data can predict the overall survival for the patient who treated with Y90 followed by sorafenib treatment. And they also analyze different clinical features and imaging, etc. Then besides that, they also analyze the lipid metabolites from the patient, and also they look at the baseline IL-6, interleukin-6 as the prognostic markers. And also they also explored the circulating DNA as the prognostic marker, etc. So given that they have been working in this area very comprehensively, then when they initially when they liaise with us and talk to both Chow and us, then so we just keep brainstorming what type of analysis we can do together. But unfortunately, because of the setup of the keynote trial, they didn't keep a lot of blood sample, and even for the blood sample, they never isolate PBMC. They only have the Buffy code DNA and the plasma sample. So since our lab has been interested in the exosome area, so we have been asking you a question: can we identify something useful from the extracellular vehicle in SORT EV, or are there any signal from Buffy code? So thankfully, with this, the paper has been reviewed, is now under revision, and hopefully in next 1 or 2 weeks' time, we can resubmit for the final revision. Then the study here is that we identify some biomarker based on the plasma EV, which could correlate with the response to SERT and sorafenib treatment very well. So thanks to this group of people, especially Dr. Mariana, who is a co-first author with me for these studies, and Prof. Cha and Prof. To and Prof. Rikus. So just a very brief introduction. So extracellular vehicle is just a very small, tiny vehicle secreted by any type of cells. So they include SMV, so which is a shield micro vehicles, and also the exosome. So in short, because we use the methodology to isolate the EV, which contain these two types of molecule. So just try to avoid the confusion. So why we keep using EV, because the methodology that we have been using can isolate both vehicles. So in our studies population, so we have been looking at this cohort. So we go back and ask the team what type of sample they still have. And unfortunately, due to the restriction or constraint of the sample availability, you can see that from the randomized phase 2 trial, under this arm is around 380 patients. But after we exclude some that are not eligible for this study, so at the end, we only have 45 patients. So we fully understand there are some limitation and bias, but we try to work within the constraint. And now we are happy to present some latest data with you that seems to be quite promising. So going back to this study population, we can see that based on the left-hand side, you can see that overall the patient median survival is 15 months. But if we based on the treatment that we apply to the patient, so you can see that there is no distinct survival benefit. So this data is similar to the entire close to 400 patient cohort analysis. So we can kind of confident that these types of studies is kind of or indirectly representing the entire cohort as well. So for the methodology, we isolate plasma, then we use a methodology called EV trap. So it's a bit-based EV isolation methodology. So we isolate the exosome or EV. We isolate the EV from the plasma, and then go through some preparation. Then at the end, we use LCMS and also with the latest models, which is provide us with the very high sensitivity for the detection. So the size of the exosome is, the EV is really following the recommendation guidelines. And we also see that the new pan EV marker MSN is highly abundant, and CD9 is abundant as well. Then after we did the proteomic analysis, we did the gene ontology enrichment analysis for the isolated EV. So again, all this data just to prove that the isolated substance, in this case EV, is really indeed they are EV. So you can see that from geo enrichment, all these are really pointing to the EV data. And if we specifically look at the global profile of the EV, we can see that it seems there is not much difference. It seems highly similar, no specific trend. But that we can we argue in the way that because there are a lot of abundant protein like CD9 or some pan EV markers, etc. So, but interestingly, if we perform the differentially expressed protein analysis here, you can see that it's really distinct to see the pattern. So the pattern becomes so distinct. You can see that on the B panel, figure B panel. The combination treatment, we can see a handful of proteins that are differently present from the patient, and then also similarly from the sorafenib treatment arm as well. And for this, actually, we purposely look at the baseline time point, so that give us the chance to look at potentially predictive biomarkers. And we do have the 6 to 9 months after the treatment time point as the comparison. but for this talk, we just present the baseline data. Then after some Venn diagram analysis, a typical bioinformatic analysis, so focusing on the SERT plus sorafenib treatment arm, so we can see that there are very distinct pattern here. So one of the protein I want to mention, but unfortunately because the paper is still under review, so for now I will just call it EV protein C. So this protein is really interesting that it's very abundant in the responder. Then according to this analysis here, you can see that it's really distinct, given that they all have a similar amount of EV markers. So, but this protein is really distinct that mostly the responder will have a high amount of these EV proteins. And the Kaplan-Meier curve, you can see that it can separate the patient very well. And the ROC curve is very significant as well. So then next, we also ask the question, is this EV protein C tumor specific, or as I mentioned, any type of cell type in the human body could secrete the EV or exosome or shield MV. So the question we keep asking ourselves is, where are those plasma EV come from? And more specific for this EV protein, would this EV protein be secreted by the immune cell, for example? So making use of the limited resources that we have, so we analyze the Buffy coat RNA. So in this case, we know that Buffy coat contains some lymphocyte, monocyte, granulocyte, and planets. So we purposely look at this, and you can see that there is no statistic significant finding for this gene C, which is equivalent to the protein C. So although it's not single cell resolution, we have no idea what type of Buffy coat will be there. I mean the composition. So, but in just a overall idea is that it seems that the protein C may not come from these cells type from the Buffy coat. Then next question is, are these proteins or these EV tumor specific? Then next, thankfully we have some FFPE tissue block remain. So we did the IFC studies. So you can see from here. So left hand side is your H&E stain, just to mark out where is the tumor and adjacent lymphatic tumor regions. And then from the consecutive section on the right hand side, you will see that this is the IFC data. So it seems it's aligned to the EV data very well, that the tissue here is quite abundant. For the protein C, the expression in this tumor, responder tumor is really abundant compared to the matched adjacent lymphatic tissue. Is apparently the tumor will have much higher expression compared to the adjacent. Then for non-responder, they are quite comparable, so we didn't see any obvious change here. But although we also aware that the sample size is quite limited, but this is really, we believe that this is a good experimentary studies. Then next, apparently we also asked, would this protein C or gene C has any prognostic value? So we just make use of the available TCGA public domain data. So we can stratify the data accordingly and do these KM plot analysis. So on the left, you can see stage 1 and 2. So there is no significant difference in terms of the overall survival. But interestingly, for this stage 3 and 4, because for the Y90 treated patient is mostly intermediate stage or locally advanced stage. So we purposely keep the stage 3 and 4 here. Then you can see that actually for this gene C, which equivalent to the protein C, there is no survival benefit as well. So from here, we can see that even the tumor will have some differences, but indeed, we can measure the difference from the EV profiles as well. And interestingly, these EV profile can be correlate well with the treatment outcome. So in this, in the first part of my presentation, I just want to summarize briefly that it just gives us an opportunity to look at plasma EV, and it seems to show that plasma EV could be a useful, non-invasive, predictive or prognostic biomarker for locally advanced HCC undergoing different treatments. Then we will believe that the tumor-specific EV could be found in plasma, and we also fully aware that the constraint of our study is that of validation cohort, sample size is quite small, and also the sample availability and also the cost of the EV proteomics. So learning from this experience, so now the post trial and the team, we have been planning for a validation studies. Hopefully we can validate the data and to prove that indeed the EV protein C has the predictive or prognostic value for this treatment cohort. Then next, I just want to touch briefly onto the immunological signals from the responder of SERT treatment. So this is the SERT treatment alone. So again, for this locally advanced HCC, as Wei Wei and others just now mentioned, actually this group of patients, unfortunately, we don't have much, we don't have the resected specimen. So in this case, for this analysis, I've been focusing on the PBMC or the circulating blood immune cells. So cut the long story short, what I have done together with the team is that we just select the responder versus non-responder, 6 versus 2, due to the sample availability. Then, and we also have 2 time point pre-Y90 treatment and 3 months after Y90 treatment. We have blood followed by PBMC isolation. Then we did the bulk RNA sequencing analysis, and we tried to look for the differences as well. And also we did the single cell RNA-seq analysis. So cut the long story short, I just briefly mentioned. So if we just look at the bulk RNA data, you can see that the responder on the right here showing that they have some pathway activated according to the gene ontology enrichment and pathway enrichment analysis. We can see that including the, for example, T cell activation pathway, and also some other interesting pathway, really abundance like immune cell activation, etc. Then if we look at multiple time points now, because eventually we have the longitudinal samples. So 3 months after the Y90 treatment, so this is the normalized data, but we can appreciate that from the responders, we can see a lot of pathway activated, for example, interferon gamma, alpha responses that are activated, seems to be quite interesting. Then next, we make use of the single cell RNA-seq methodology. So we just pick one responder and non-responder based on the sample availability again, then 2 time point. So this is the single cell RNA-seq data. You will see that blue color indicating the baseline of the sample, and the red color indicate the post-treatment time point. Then based on this, we did a graph-based classification. We can easily identify 17 cluster, etc. But cut the long story short and just keep it brief. So we focus on CD8 T cells. So you can see that there are different numbers of the CD8 T cell can be found from this patient PBMC. And interestingly, if I zoom into these CD8 T cells, and then I ask a question, are there any specific TCL usage, or after the Y90 treatment, are there any specific TCL expansion being found? So interesting, here after the analysis, I can see that from this particular responder, we can see that there is one particular alpha chain and beta chain TCL being expanded, or they have the priority being used. So you can see from the Violin plot here. So the responder have expanded clones from these T cell genes, and unfortunately, comparatively from the non-responder, they don't. So apparently, these 2 patients, the immune cell or the CD8 T cell, they are using different TCR. But from here, just the exercise to show that there are unique T cell clone being expanded, or is being the kind of priority being used by the T cells. So if we zoom into this type of T cells, then you will appreciate in the way that mostly the responder will have these T cell clones. And after the treatment, these T cell clones are being expanded as well. Then we ask another question, what is the pathway, etc. So also cut the long story short, you can see here is that the T cell after the treatment have a lot of interferon gamma signaling. But before the treatment, we always see the TNF alpha signaling via the endopalpy pathway. So interestingly, this T cell data is similar to the bulk RNA data that we observed, although bulk RNA sequencing data lack of the single cell resolution, but we can see similarity in the way that the interferon gamma is being upregulated after the Y90 treatment in the responder. So then it seems it's really interesting to see this. We will further this analysis in the future. And last, I just want to show you some preliminary analysis that we also try to use some spatial transcriptomic methodology to study the Y90 treated patient. So it's just an example here. So we have the Y90 treatment followed by atezolizumab, anti-PD-L1 and anti-VEGF treatment, and also right-hand side is a treatment naive from another patient. So it's just a idea to give you that the CD45 is to mark the pan immune cells. So we can see that after the Y90 treatment and atezolizumab, that there are a lot of immune infiltration, and according to gene expression analysis, you can see a lot of different immune-related pathway genes are regulated, like B cell and antigen presentation cell expression and macrophage markers. They are highly expressed as well. So in summary here, so I just want to showcase that the search responder has quite unique T cell usage and expansion in the PBMC. Even we, unfortunately for this group of patients, we don't have resected specimen, but we can still extract some information from the PBMC. And single cell RNA-seq and RNA-seq can show some similar immune activation signal. So if and also spatial transcriptomics is quite useful to investigate in situ immune activation. So I would like to stop here, and thanks again for the opportunities for me to share this now ongoing studies. And again, all the data are unpublished and is in progress. So I hope to present a more concrete story next time we meet, hopefully face to face. So last, I just want to thanks again for the trial group invitation, Prof. Chow and the teams, and my NAP in NCC under Prof. Toes, and also all the surgeon and physician, and also more importantly, our collaborator from Germany. So I'm happy to take any question. Thank you so much.